Good afternoon, distinguished guests. Welcome to the NTU College of Medicine Dean's Lecture. To commence the event, please extend a warm welcome to Dr. Mingxian Wu, Dean of College of Medicine, National Taiwan University, for the opening remarks.
Our distinguished guests, Professor Hirsch and Professor Losman, dear colleagues, ladies and gentlemen, good afternoon. On behalf of the College of Medicine, National Taiwan University, I would like to express my sincere welcome to all of you to the NTU College of Medicine lectureship and our Nobel Prize lectureship. We'd like to express my thank you for Song Gongyan Foundation to provide the support of these two very important lectureship, especially in the advance of neuroscience. These two lectures are so important that deserve everybody to hear, to learn the recent advance in neuroscience. I would like to thank you, our two distinguished guests came to visit our College of Medicine, and Dr. Rosman also leave his name signature in our chair at our faculty club at College of Medicine. Thank you very much, and welcome all of you once again. Thank you.
Thank you, Dr. Liu, for very warm remarks. Now, please welcome stage Dr. Lin Zongzhen, Associate Professor in Graduate Institute of Brain and Mind Sciences at the College of Medicine, National Taiwan University.
Good afternoon, everyone. My name is Lin Zongzhen from the Graduate Institute of Brain and Mind Sciences. Welcome to the Dean's Lecture. Today we are honored to have with us Professor Joey Hirsch, a leading pioneer in human brain imaging and social neuroscience. Professor Hirsch is Elizabeth Mears and House Jameson Professor of Psychiatry, Comparative Medicine, and Neuroscience at the Yale School of Medicine. She also holds appointment at University College London and serves as senior investigator at Huskin Laboratory. Her distinguished career spans decades across Yale Memorial Sloan Kettering Cancer Center and Columbia University, where she founded the Functional Imaging Research Center. She made seminal contribution to functional neuroimaging, first using fMRI to map visual perception, cognitive control, emotion, and language, and now leads two-person neuroscience, observing two brains synchronized in real time using innovative FNIRS hyperscanning. Her research has revealed fundamental principles of how social interactions are represented in the brain. This insight helps bridge neuroscience and clinical practice, enhancing understanding of social dysfunction in neuropsychiatric disorder as well as social condition in healthcare. She has received numerous honors, including the George Gamow Distinguished Scientist Award and a recognition as a top woman scientist by the World Science Festival. Please join me in welcoming Professor Hirsch, who will present the new neuroscience of two, breakthrough neuroimaging technology and machine learning for understanding human social behavior in the live and natural world.
Very good. Thank you so much for that kind and generous introduction. It is such an honor to be here. Jim and I appreciate so much the accommodations that you've all made, the organizing committee for inviting me to give this lecture. It's truly an honor to be here. And I'm very excited to tell you about some of the new domains in neuroscience, particularly systems neuroscience, the kind of neuroscience where we actually study live human beings. You know, not the kind of neuroscience where we study the squishy little neurons. This is the real stuff. This is the stuff about the relationship between neurocircuitry and behavior of human beings. And I'm very fortunate to have been in a position to start a new kind of neuroscience, and that is the neuroscience of two, is what I refer to, but the neuroscience that relates to neurocircuitry of two people interacting in the real world. So, starting with the self-evident, humans are extraordinarily, profoundly social. And this is something that we all know. From the beginning of our lives, all the way through our lives, we spend time with each other. Probably the most of our time and our entire lives are spent in interactions with other people. And one of the really important venues for social interaction is the human face. And faces are some of the most important stimuli that we have as human beings. And so in the study of two persons neuroscience and in the quest for understanding the basic mechanisms of social interactions, we start with the human face. We have organized the human brain in lots of different ways. Originally, it was organized by Brodmann in terms of its cytoarchitectural areas, and there are 52 different cytoarchitectural types in the brain, suggesting a multiplicity of functions. We also organized the brain, of course, by its anatomical features, and this is all well known. The human brain and its anatomy has been studied for decades. We also organize the human brain by its functional parts. And this is the main mission of systems neuroscience, is to understand what parts of the brain do specific things. And we have this notion that specific parts of the brain are involved in specific functions. And in fact, that idea has been the idea that has actually fueled an awful lot of our social neuroscience. Basic neuroscience, not just social neuroscience, but neuroscience in general. And so the quest of understanding how the different parts of the brain do what they do is really a main mission of neuroscience. Now, most of what we know about the function of the brain recently has come from functional MRI. And functional MRI is an absolutely revolutionary technique, because what we can do is put typical normal brains into a scanner, ask people to do things, and measure the brain activity that's associated with those tasks. And so here's an illustration of somebody going into the scanner, and you can see as someone goes into the scanner what a solo task it is. We put somebody into the bore and we ask them to do a cognitive task, and indeed we can learn a great deal about how the brain processes cognitive events. The basis of this is what we call the hemodynamic response function. And that is a basic principle of the brain, that wherever there's active neural tissue, there is a recruitment of highly oxygenated blood. That is called the hemodynamic response function. And no matter how we measure it, whether it's with functional MRI, which is measured, the hemodynamic response function is measured by magnetic susceptibility, or whether we use optical properties, that is to measure the oxygen concentrations of the blood that's actually recruited. No matter how we measure it, it is that signal that tells us about the brain. So we put someone in the scanner, we ask them to do things, we measure the hemodynamic response function, and we can actually learn a great deal about the brain. Now, as I said before, the face has been a very important stimulus in understanding both cognitive processes, visual perception, and social processes. The face is probably the most important stimulus in all of the repertoire of stimuli of human beings. And so, in the original studies using a scanner, we learned a great deal about the human face. And so we can put someone in the scanner and we can show them pictures of faces and objects and faces and objects and faces and objects. And what we learn is that what we consider the ventral stream of the human brain. Now, for those of you, probably everybody here is really familiar with brains. Top of the brain, front of the brain, back of the brain, bottom of the brain. This is called the ventral stream because it has to do with the ventral part of the brain. What we know is that people's the neural circuitry that's associated with faces, as seen in the scanner, shown by pictures, really activates a whole lot of territory. We refer to this as the real estate principle associated with the human face. There's a ground truth associated with that. Whenever, as scientists, whenever we have a major finding, we usually like to replicate it. And of course, this finding is well associated with single unit recordings with monkeys. And again, face processing, face specificity is the job of the entire ventral stream of primates. And so we consider that evidence that indeed that what we see in magnetic resonance imaging with humans, we also see with primates. We consider that a ground truth. But there's a significant knowledge gap in the prior knowledge of social neuroscience, and that it's based on single individuals with simulated pictures or simulated stimuli. Most of what we do in our lives, day in and day out, is live interactions with other people. And this is really not possible in the scanner. And so the truth of the matter is, in the mainstream of social neuroscience, we know very little about the neural circuitry that's associated with live interpersonal interactions day to day. And I bet you guys could guess that it's probably pretty different, that the brain is organized in very unique and systematic ways to connect with other people, and yet that remains an unknown frontier. And indeed, that's what I want to talk about today. So let's unpack that just a little bit. Start with, what in the world is a social interaction? What goes into a social interaction? So let's just take this simple case. Two people are sitting across from each other. They're talking to each other. Messages are sent. The message is sent by either facial expressions, by voice. It's received. Another message is then sent in response, so a reciprocal message is sent by face and voice. It is indeed received. And what we have is a dyadic cycle that goes on in a reciprocity. Oftentimes it happens very rapidly in social interactions. So you can see a social interaction, a single simple social interaction, is a very, very complicated process that is our mission to try to understand. So let's move on. One of the reasons why we want to understand this is that should we ever want to design a pseudo-human robot-to-human interaction, we'd really like to understand what in the world we need to do to design this little guy here to actually send credible messages to this guy over here who's perhaps receiving some kind of therapy, and superimposing what we know it goes on in the paradigm. There's facial information, there's voice information, and so on. Then this guy receives this information, and then he sends something back to the robot, and so on. We would like to understand how we could build some kind of simulated interaction if we really understood how it worked. So what do we need to know? All right, so first of all, why is this important? Well, first, that socialness, this is a fundamental component of our survival, and we really don't know very much about it. And one of the problems here is that we've got a loneliness epidemic, and we might understand a little bit more how to resolve that if indeed we had more information about the underlying neurophysiology that connects us to each other. Many clinical applications of social disorders such as autism disorder, schizophrenia, depression, bipolar disorders have a very, very strong social component, and it's that social component that oftentimes is not well understood, and yet it's the behavioral aspects are used diagnostically, and a stronger neurophysiological understanding of those conditions would be beneficial clinically. And then finally, the example that I showed you, the development of brain-inspired neural models of robot-human communication would benefit enormously from having a model of actually how it's done in the live human being. And so that brings me to the roadmap of my talk, The first thing I'm going to tell you about is a paradigm shift in neuroimaging that allows us to actually measure live interactions. I mean, one of the reasons why we don't know very much about this is that we haven't had the equipment, the technology, the tools to actually do the measurements credibly. And so I'm looking forward to sharing that with you. I'm going to show you evidence that convinces me that their neurospecificity for live faces and visual interactions is very specific to the live case. And of course, as scientists, we develop models. We'll start a model of the hierarchical processes that are associated with live interactions. I'm not going to stop with faces. I want to bring on speech, and we'll talk about the similar building of speech models on top of face processing, and then finally a grand model of constructing a comprehensive brain-inspired large neural model for social human interactions. So here we go. Paradigm shift from the scanner to a surface-mounted technique called near-infrared spectroscopy that actually allows us to do brain imaging of two people under simultaneous conditions. So here we are with an example, a head-mounted, surface-mounted detector device. And so our participants wear a helmet of detectors, and these detectors are actually both detectors and emitters of light, which I'll show you in just a minute. I think we haven't skipped a slide here. But these head-mounted detectors allow two people to interact with each other. We're actually measuring the hemodynamic response function by absorption of light, which I'll show you in just a minute. Let me just tell you a little bit about the advantages here. First, it's tolerant of limited head movement. So the participants can be upright, they can be talking to each other, they can be in natural situations. Our temporal resolution is very, very good. We're measuring at about 30 hertz per signal, which is amazingly rapid. Our spatial resolution is quite terrible. Related to functional MRI. And this is the trade-off here. In order to understand, in order to measure neural activity through optical methods in live conditions, we have to sacrifice the high resolution of the MRI. It's worth it in order to get the measurement of two people simultaneously, but this is the sacrifice. Our resolution is about 3 centimeters. It's 3 centimeters on the surface. It's also 3 centimeters in depth. And so anything that I tell you today and anything that anybody tells you about measurements from near-infrared spectroscopy, it's the superficial brain. The sweet spot of emotional systems from the center of the brain are not accessed with this technique. And so that is the disadvantage. We're also not collecting the neuroanatomy, the actual anatomy of the brain. With fMRI, the neuroanatomy comes as a freebie. But here we make an assumption about the underlying neuroanatomy based on models from EEG. But one of the great advantages here is that we do not have a high magnetic field. And so what we're able to do is add quite a large number of additional measurements simultaneously. So here you can see the lines here. We're imaging. We're not only doing the optical imaging here, the near-infrared spectroscopy, but we're measuring EEG simultaneously. We're also measuring eye tracking, scene cameras, facial classifications, behavioral measures, physiological measures. All these things can be acquired simultaneously on the same time frame, and so what we achieve is the acquisition of a bundle of images, a bundle of measurements of which we can use to understand our images in a different way. Now, basically, this is not magic. It works by, as I indicated before, light absorption. And the basic principle, again, is the hemodynamic response function. We're high levels of oxygenated blood are recruited to active neural tissue. And so it's our job with our optical imaging techniques here to measure or identify the areas in the brain that are actually recruiting oxygen, and that's our proxy for neural activity. And so we have emitters in the cap. This is a coronal view of the brain, and you can imagine the cap that I just showed you. And the optodes are divided between emitters and detectors. The emitters actually inject light into the brain of specific wavelengths. In this case, 780 nanometers and 830 nanometers, responsive to deoxyhemoglobin and oxyhemoglobin. We know how much goes in, and we know how much comes out. And then, based on what is absorbed, the difference between what goes in and what goes out is our indication that there has been a recruitment of highly oxygenated blood to that area. And so basically, that's how it works. This is the absorption table and the functions here that identify the reasons that we've chosen those particular wavelengths. Okay, so this is just an example of kind of how it works. This is somebody wearing one of the near-infrared spectroscopy caps, and I'm pointing at one very specific detector. This is one detector, one person, and one run. This is as raw as it gets. The task is finger-thumb tapping. And this is a fiducial task that we use to make sure that we're doing what we think we're doing. The time series here is that we ask the person to tap their fingers for 15 seconds and then rest. Tap, rest, tap, rest. This is a 15-second block design. And what we see is, in the case of the oxygen goes up when, in that particular optode, when the tapping is occurring. It falls during rest, up during the task, falls during rest, and so on. That's the oxyhemoglobin signal. The deoxyhemoglobin signal is correlated to it. We actually use both signals in our analyses. So that's basically the bottom line of the signal that is used to measure neural activity from these live caps that people wear during these interactions. So, okay, so this is not a new technology. It was discovered around 1997 by people that Jobsis and Wyatt, who actually discovered that one could measure oxygenation in the blood using near-infrared spectroscopy. And it wasn't until 1993 that Britton Chance and others discovered that one could actually measure task-related activations in the brain. And that was around the time that Sezio Gaba made the discoveries for functional MRI as well. So this was the task-related indications of changes of the oxygenation in the blood of the brain during activation was discovered around the same time. And you can see that it was a little bit slow to take off, but more recently the technique has reached much more use in the field. So let's start at the very beginning and ask, does it really matter that we look at live faces as opposed to, say, simulated faces as we have understood them, the neural activity associated from functional MRI? This is an example of how we actually would ask and answer that question. This is an example of the setup. Two people are not wearing caps here for illustration purposes, but I want to point out this screen in between them. This screen is what we call a smart glass. And there's sometimes the smart glass is open and people can see each other, and sometimes the smart glass is closed. We run a current through it and it becomes opaque. And due to the smart glass, we can time when people are actually looking at each other or interacting. The time series is like this. For 15 seconds, they can look at each other, and then there's a rest, and then they look at each other, and there's a rest. And I can break up the times when they're actually looking at each other. And I'll show you from time to time, that's an important thing to do from a paradigm point of view. But the smart glass is very important because I can time the behaviors to the hemodynamic response function of the brain. So I've adapted the experimental protocol to the signal in the brain as we know and understand it. I just want to point out that all the equipment that's hanging from the ceiling mostly, these are face cameras, eye tracking. Eye tracking is mounted on the smart glass, and we can do all the measurements we need to do of the ocular motor system simultaneously with the face looking. So this is our general paradigm to understand face processing system. Okay. Does it matter if the face gaze is live? And the way we understand this is that we have people directly looking at each other along the paradigm that I just showed you, and we can compare that with, in this case, a video face, simulated face, but it moves. And this is a very traditional face that's been used for studying in the scanner. And what do you think? If one actually compares the real face with the video face, one can see that this large patch of brain, this is right side of the brain, supramarginal gyrus, is in what other people have referred to as a nexus of social activity, and here it is. This is a huge patch of brain that's much more interested in the gaze at the partner's face than the gaze at the video case. So yes, that it really does matter that the face is live. Okay. So we never take one experiment as being an answer to a big question like this. And so in this group of experiments, we compared live face gaze with the face of a three-dimensional robot. This is Maki. He's a pretty smart little robot. He moves his eyes, he moves his face, he moves around. He has very human-like features, but he's clearly, obviously, not human. And indeed, the brain, you can't fool the human brain. The human brain has a much more exaggerated response to the live face than to the robot face. So again, we have an answer that yes, the brain is much more responsive to live faces. But then the next question arises, what in the world is this supramarginal gyrus doing? Why is it so much more responsive to the live face than the simulated face? What's the difference? Well, one hypothesis is that the supramarginal gyrus is specialized for encoding of facial expressions. Well, we can address that question. We know a lot about how to partition facial expressions. In fact, there's a whole specialty in AI that looks at faces and identifies specific configurations of the face, not having nothing to do with the emotional content of the facial expression, but just the facial expression itself, and they can be quantified. And as cameras are looking at faces in real time, they can identify the amount of time that the face is actually associated with each of these individual facial expressions. So we set up an experimental paradigm that can actually measure facial expressions. So we have two people, a face watcher and somebody who is an expression generator, and he's just looking at funny or kind of yucky movies. Anything that actually is designed to generate a natural, honest expression. Sometimes they're funny little animals playing with each other, or sometimes it's yucky spiders and the expression generator goes, ooh! And sometimes it's just a happy expression, but whatever it is, it's a genuine expression. It's associated with something that is being seen. What we do is take the live facial expressions that are acquired by the camera and we run them as regressors on the neural responses of the individual, that is the face watcher. And when we do that, then we can look at the brain of the face watcher in response to the facial expressions. And when we do that, we see that the supramarginal gyrus is incredibly interested in facial expressions. And so we've learned something very important about the specificity of the supramarginal gyrus and live faces. But there's lots of other things that could be responsible for why this area of the brain is interested in live faces. And one of the reasons might be that maybe the ocular motor system, that the eye movement system is actually gathering information about faces, and that is also integrated into this area. So it's not just that the perception part of the brain is interested in faces, but also the ocular motor system that's integrated with it. So it's a very reasonable hypothesis, and we can go about testing the reasonable hypothesis. First of all, we know a little bit about the exploratory movements of the eye, saccades and fixations, and any of you who are vision scientists out there know that the ocular motor system has been studied extensively, and we can track the visual pursuit movements in terms of saccades and fixations of any kind of exploratory mission of a stimulus, and indeed we do that with faces. And particularly, we're interested in fixation duration because we're interested in how long the eye actually stays on any one place in the face as an indicator of how much information about the face is being sent upstream to the perceptual systems. So the saccades are the sort of the delivery system for the fixations, and so that's just saccades and fixations are really the primary dynamics of the ocular motor system that we're interested in. Particularly, I'm interested in fixation duration as it relates to the face processing, and that's what I'm going to show you now here. And this is how we measure it. Again, we have a face watcher watching somebody's just live face, and we measure the fixation duration with our eye tracking system, and then we use that as a regressor on the brain of the neural responses. And what we see is activity that is extremely robust. Again, the supramarginal gyrus, but also the ocular motor system involving much of the sensory motor systems associated with face. And so again, there's a robust sort of yes answer to yes, it is true that the ocular motor system is involved in the part of the brain that is very specialized for the live face and not for the simulated face. All right, so this brings us really to an opportunity to begin to construct a model. We know that the simulated face, the social system, is associated with the ventral stream, but as we add to our understanding based on the live face, we see that we're moving upstream to the supramarginal gyrus, and again involving the entire ocular motor system. So we're progressing up the brain in terms of hierarchical processing that's associated with the live face, which is pretty exciting. So there's other things that are associated with social interaction, not just for appreciation of the live face. Eye-to-eye contact. Eye-to-eye contact is an extraordinarily intimate social behavior. And it really has never been studied before under live conditions, and we have the opportunity to ask how these brains are actually responding to eye-to-eye contact, and indeed, is it related to the activity that is associated with the visual perception of the live face. And so we go about to ask the next question having to do with eye-to-eye contact. And again, using our paradigm, we can measure eye-to-eye contact. We sort of make a little rule when people are looking at each other. We have these eye boxes that we just computationally put on the subjects of each of our subjects. And we make a little rule that whenever each person's eye gaze is in the other person's eye box, then, and this lasts for 10 frames, we call that a one eye contact event, and that's about 0.83 seconds. So we measure how many of these events occur during the 3 seconds of the 3 seconds of looking. And here, this is very complicated. It's too complicated to understand, but we have eye tracking for one person, eye tracking for the other person. These are the times when the eye tracking occurs. And the green bar is when everybody is in each other's eye box. And so if we blew that up, there would be many, many dots in those areas indicating how many eye contacts there were in each of those, in this case, 3-second opportunities. So we use that also as a regressor and ask, how is the brain responding to these live eye-to-eye contacts? And the first thing you notice is that it's not the supramarginal gyrus, that eye-to-eye contact involves a very different part of the brain. We call this the dorsal stream, the top of the brain. Now, vision scientists know a lot about the dorsal stream. It's a part of the brain that's associated with attention, with eye movements, with purposeful direction of gaze. And so it's not surprising that the social aspects of eye-to-eye contact would also involve some of these same areas. Also, the dorsolateral prefrontal cortex is always there. It's sort of the control mechanism. It doesn't care what the brain is doing. As long as it's doing something, the dorsolateral prefrontal cortex is involved. So anyway, the dorsal stream is the focus for eye-to-eye contact. And this is a major discovery in the field that that eye-to-eye contact involves areas that are separate from just the face processing system. So the important part of this finding is that we really need to extend our model, and instead of just the ventral stream and the lateral stream associated with face perception, face processing, and ocular motor system, we actually now have to add another part of the whole social process associated with a very intimate face-to-face gesture, eye-to-eye contact. So we're beginning to understand a lot more about the social system of the brain. And so the idea here is that the more complex the social system gets, the higher up we seem to be going in the brain. So let's just sort of test that model as we move from face-to-face system to the voice or the language system. Much of what we do when we communicate with each other is that we talk. We don't just look at each other, we talk to each other. And so how does the talking in interaction differ from what we've learned about the canonical language systems in the brain? So here, we will start with that. First of all, what we know about the canonical language systems of the brain, we've learned from MRI. We put people in the scanner and we show them pictures, and we ask them to silently to themselves name those pictures. And indeed, when that happens, what we're able to do is identify the canonical language systems, Wernicke's area for speech understanding, and for speech production, Broca's area, and so on. So this is the canonical language area. We can see them all the time in the scanner just by asking people not to actually speak because movement is not allowed in the scanner, but to actually just think what they would say if they were just naming and describing a picture. It's a classical task. But there is a ground truth associated with that. We know that we get the right answer because when we map the language systems of the brain, we can map them for physicians for neurosurgical planning. And so if a physician is going to do surgery on a brain, and he wants to protect the eloquent or the functional tissue, needs to know where the language systems are, the sensory systems are, and so on, so that there's not undue damage that occurs to the surgery. So in the case of a surgery that might put at risk the language systems, we map the Broca's area, Wernicke's area, and so on. And in the process of doing that, the surgeon will always validate the map with the interoperative cortical stimulation. So we always, for language maps, have validation based on the interoperative cortical stimulation in the OR of human beings. And when that occurs, then we have a very, very strong indication that the maps, as we know them and as we obtain them with functional MRI, are indeed there. So the question is, do we actually get those maps when we do a two-person, actually live mapping process? And indeed, we set up this interesting paradigm where we have two people, as we have here, and they can talk and listen, sharing the listening and the talking, and doing the same picture naming and description task. And when we ask people to do that, sure enough, we actually obtain the canonical language areas: Wernicke's area, Broca's area, and the articulatory system having to do with speaking and with listening, the receptive part of speech. This is true when we use the deoxyhemoglobin signal. It's sort of a detail of doing the optical imaging. The oxyhemoglobin signal is too confounded because of the change in the oxygenation due to speaking that it doesn't give a valid result. But the deoxyhemoglobin signal is a perfectly good signal to use for the language. So this validates what we know to be true and gives us a green light to go ahead and use the near-infrared spectroscopy as a tool to study interaction in person-to-person language. So we move forward with this. We ask, does interaction in language matter? That means, is your brain different when you're talking to your friend who's standing in front of you, or if you're talking to yourself in the morning in the mirror, whatever. So does interaction matter when you're talking? And the answer is, indeed, just as you would suspect, it does. It matters a lot in terms of the receptive part of language. This is the non-interactive and the interactive language. It appears it doesn't matter so much with your speaking. The speaking system seems to not care if you're talking to anybody else, but the receptive parts of the brain matter. It matters a great deal. So again, your brain is very different when you're talking to somebody in real life than when you're just talking to yourself. Or it's very different than the kind of imaging, the kind of language system we've seen as we image in the scanner. So the evidence for this fact is also based on functional connectivity, or studies of dialogue and monologue, and we see that the direction of the mechanisms is also bidirectional during interaction and unidirectional during a monologue. So the brain, again, evidence the brain is very different under interactive, social interactive conditions as opposed to not. Okay, this is sort of the crowning experiment then. What happens in a less constrained environment where you have two people together, they're talking to each other, they're looking at each other's faces, and they're agreeing and they're disagreeing. Now, this is an experimental paradigm that uses all of the systems that I've talked about and puts them all together under one interaction, and we ask the condition, how different are people's brains when they're under conditions of talking to each other under agreement and talking to each other when they're disagreeing. And so we were able in this experiment to bring people together with different organic agreements and disagreements on topics that we tailored to our individual partners. And in this experiment, we had people talking and listening in about 15-second epics, as is typical of our two-person paradigm, and asked them to do this experiment under conditions where they were discussing things that they agreed and disagreed. And our question was, how different is the brain under these conditions? Is it just that the canonical language systems and the facial action unit systems are different, or does it all mush together in some unspecified way? And indeed, that was the finding. Under these complicated social conditions, and simply talking and listening to each other with agreements and disagreements, we see the social systems very partitioned for agreement, executive systems, frontal brain, very isolated for disagreement conditions, and we see a whole different picture of the brain depending on the social interaction and what the two partners are, how they're relating, and exactly what we're doing. And so this, I think, is the beginning of the real challenge for this new field of the new neuroscience of two, is understanding how these many single mechanisms are linked into a working interaction unit. And so I think one of the things that we begin to see here is how complicated and how simplified it is under conditions of social interaction to see these mechanisms of the brain come together. Okay. So that sort of brings us back to the beginning with the question of, if we really wanted to design one of these guys to be a companion to a live and interacting human being, what would we need to do and how would we do it? Have studies of this kind taught us anything about how we would need to set up this model? What have we learned that could guide us in this task? Well, the first thing that we learn is that live human faces are primary sources of social information. Now, if that's the case, we need to do something about his makeup, because this is not a very expressive face. And so in a responsive kind of dyadic interaction, we might want to imagine that we could build responses that were not just language, but responses that were also gestural and perhaps suggestive of facial expressions. Secondly, that the algorithms that we use to encode the information from our participant here probably need to be designed in a very efficient hierarchical manner, and I have to say that we have a lot to learn in that respect, as well as we put together information from our participant to, in this case, our robot. That the neural mechanisms of the live face need to probably include not just the face, but also the eye mechanisms, the whole oculomotor system, and how the information is gathered could be one of the ways in which this robot could be made a lot more efficient. And the visual fixations actually need to be related in some way to the actual features of the face. It would probably be very important in connecting the dyadic interactions between the two participants here. Finally, I think that in a dyadic situation, when eye-to-eye contact triggers specialized social actions, that it requires a very, very sophisticated response mechanism associated with our robot and our human. So there are many cues from our human that the robot needs to be responsive to, and indeed he needs to send signals back. So this is a complicated and daunting task, and I think that it's work like this that begins to identify what we need to think about in order to set something up like this that works. And so I think with that I will end and thank the many people from my lab who's helped bring together all of the parts of this work over a long period of time. And thank you.
Thank you, Dr. Hirsch, for such an informative speech. Now we enter the Q&A session. Please welcome Dr. Minzong Zheng back on stage for the Q&A session.
Hi, this is Jason Tai from the NTU Vet School. So myself is a vet. I'm just thinking about, because what you just mentioned are about, for example, the eye contact or the facial recognition. But sometimes interactions are more complex. For example, like my gesture, my posture, and even the physical contact could make difference. So is there any chance that your study also include these kind of extra factors that will affect the reactions afterwards?
That's, I think it's a really, really important question. It first builds on the assumption that these social interactions involve many, many social cues. And in the work that we've done, we've controlled only a small portion of them. But body gesture, facial expressions, many things, just jittering, just moving around, all of these things are social cues that are, one, received, and it's interestingly enough, people that you're in a dyadic interaction with, they respond to. And there's this wonderful social phenomenon called mimicry, where if one person is moving in a particular way, another person's movements will also be more synchronous. One of the things I didn't talk about was neural synchrony. In interactive conditions where there's a lot of co-responsiveness going on, that the brain responses become quite synchronous as well. So you pointed out that there's many, many more complexities and variables in this system than what I pointed out. It's a very new field. I feel very fortunate to have been to be very much at the headwaters of it, but you can see how much more there is to understand. We just barely touched the surface.
Sorry, I have an evil kind of idea. I know it's definitely for human benefit study, but when I see the result of the agreeable and disagreeable, the region of the brain, would it be a target for the future manipulation for human?
I love that question. For any of you who didn't understand, the question is, does the study of agreement and disagreement and looking at the neural correlates of those different attitudes tell us anything that could be relevant to, say, world peace or something really important like interpersonal interactions and conflict resolution? Is there something here? Does the biology actually inform us in any way that could help us improve negotiations between warring parties? Gee, I hope so. But I can't help but see that there might be some relevance. Is that if we understand how the brain actually manages these two states, could we actually use that to our benefit? One of the things about the results that was so surprising to me is how different they are. I would think, in fact, I predicted, and oftentimes my predictions are wrong when we start an experiment, but I literally predicted that the brain has its well-developed systems. We have a language system, we have a face processing system, we know how they work, and they're going to work pretty much the same way, whether you're agreeing with your partner or you're not. Well, wrong. That's not the case. The brain fooled us all that it's very, very different. It combines these systems in a way that was unpredictable before we did these experiments. That agreement and disagreement are very different states of mind. And these different states of mind have different neural circuitries associated with them. There's a lot to learn there yet. Very good question, thank you.
Professor Hirsch, thank you very much for your inspiring talk. May I ask one scenario that's recently more and more common in our research environment as well as teaching environment, that is online meeting. So for this kind of meeting, we asked our colleagues or our students to turn on the camera, and we thought that there's some kind of eye contact, but how does that kind of eye contact affect our neurocircuitry? Thank you.
That's a very, very good question. And I actually didn't talk about that in this talk, but we last year published a paper where we did the experiment with again, face processing live versus the same face processing on Zoom. Thinking that it's exactly the same face, the same features, everything's the same, so we should see the same results. Wrong. The results were extremely different. The difference was that this supramarginal gyrus was not active. Well, not active. This is a relative thing. It was less active than in the real face condition, and the eye movements were very different. And it's suggestive of what we think is that the Zoom stimulus is a sparse stimulus. That is, we don't have the same spatial or temporal resolution on the screen that you have in real person. And furthermore, the eye contact is not direct. That is, it's at an angle. And so whatever the brain's mechanism is for receiving and responding to live eye contact, it is degenerate in the Zoom condition. This is just me thinking about it, but one interpretation I have for the difference is that one of the reasons why people are tired after a Zoom conference is that the stimulus they're receiving is so sparse, you have to work harder to get the information. It's more energy requiring. That's just an idea. That's not what the data say. The data just say it's different. But it's a really, really good question, and it's a really important question for the kind of world that we live in, where we teach and we do our negotiations so much by Zoom. And it's exactly, it's not at all the same social experience as face-to-face. So, thank you for that question. It's a very good one.
Okay, due to the time constraint, we have to close this section. So please join me in thank Professor Hirsch for a wonderful talk today.